Chapter 25, we're going to be looking at the history of life on Earth. One thing that I want to make very clear right now is that life on our planet, according to the theories, came from non-living things. And it was illustrated that this is possible through a series of recreated events of how this actually could be done. This is serving as evidence that life could have happened just by chance, naturally, through changing events in our global history. With that in mind, I'll probably reiterate that at some point that a lot of our evidence has been proven to be possible through experimentation. But an overview first, Lost Worlds. Thinking back about what you know about Antarctica and the environment, you know that it's very, very cold there, and it's very hard for even humans to exist there. Of all the living things that live there full time, meaning year-round, the largest animal is a fly that grows up to only be 5 millimeters in length. Everything else that's bigger than that, they might live there for a period of the year, but they don't live there year-round. With this in mind, researchers and explorers were quite startled when they discovered very, very large dinosaur fossils dating back to 500 million years ago. How is it that these reptiles could have existed there when the largest thing now is only 5 millimeters and it's very, very cold? Well, at this time, 500 million years ago, the ocean waters surrounding Antarctica were actually quite warm, and they were teeming with tropical invertebrates. So the fossil record says. Later, these lands were covered in forests. Geographically, looking at other parts of the world, we see the same thing going on. Past organisms found there are very different from the ones that we're seeing in those same places today. And this illustrates the pattern of macroevolution, which is where life in general is evolving much more than just one species. Life is evolving. Examples of this macroevolutionary change: the emergence of terrestrial vertebrates through a series of speciation events, the impact of mass extinctions on the diversity of life, and the origin of key adaptations, such as flight in birds. So, conditions on early Earth made the origin of life possible. These four things are what we hypothesize to have happened, and in this sequence, in order to develop life. Now, what I want to start once again by reiterating, each one of these four things has been experimented and tested and found that it can actually happen. These have been recreated in a lab setting. The first, abiotic synthesis of small organic molecules. And we're talking about amino acids here and the nitrogenous bases that are found in RNA and in DNA. So, without any life at all, these molecules are able to be built. Secondly, the joining of those small molecules, amino acids, into larger macromolecules. And we know amino acids combine to form proteins, and the nitrogenous bases, A, T, C, and G, and if we're talking RNA, U, can combine to form nucleic acids. That these proteins and these nucleic acids can package into protocells, and these protocells are droplets with membranes that maintain an internal chemistry different from that of their surrounding. In other words, they maintained a level of homeostasis. And four, the origin of self-replicating molecules that eventually made inheritance possible. Evidence suggests that Earth was formed about 4.6 billion years ago. At this time, the atmosphere that existed was thick with water vapor and other elements, nitrogen, carbon dioxide, methane, ammonia, hydrogen sulfide. As Earth cooled, the water condensed, and that created the oceans that we have today. It was back in the 1920s that the idea of organic compounds arising from simpler ones could have happened. Organic compounds are carbon-based, so those would be the macromolecules we looked at way back in like chapter 2, including proteins. This was proposed by A.I. Aperon and Haldane. They hypothesized that Earth's early atmosphere was reducing, and recall that means that we're adding electrons if something's being reduced. Reduced oxidation. The energy to synthesize these compounds very likely came from lightning and intense UV radiation. Keep in mind that the UV radiation from the sun back then would have been much, much greater than it is today, as our atmosphere has significantly changed over the course of those 4.6 billion years. We now have a good layer of protection in our ozone layer, which didn't exist back then. They also suggested that the oceans were a solution of organic molecules from which life arose, and they called this an organic soup, I believe. As suggested, the first organic compounds on Earth very, very likely occurred near deep-sea vents. A lot of heat there, there's submerged volcanoes down there, and there was a lot of those organic compounds that could be assembled. And in this image we see a recreation of that possibly happening, and we found that yes, it very well could have, as illustrated by our experiments. Stanley Miller was a pretty big name, by the way. That's one you're going to want to remember. Miller constructed an apparatus to simulate the Aperon-Haldane hypothesis, and we can see that right here. We can see that there's a little container representing the atmosphere with a bunch of those compounds that we believe were in the atmosphere in early Earth history. We also have an image of him simulating lightning strikes, causing these to rearrange or reconfigure themselves, and with the water vapor that could condense and cool. We see little condenser coils there cooling the water vapor. We have the formation of oceans. We can also see that he applied a heat source to represent those hydrothermic vents and deep-sea volcanoes for the heat source. And what he found is that the amino acids found in living things, along with other organic matter, was able to form through this procedure. So once again, a lot of our notions and ideas of how life could have arose from non-living things were recreated and found that they really could have happened. One of the more recent illustrations was a synthesis of RNA, and that's what we see over here. Could macromolecules such as enzymes and other proteins, like this nucleic acid, have formed on early Earth? Because this is what we need if we're going to self-replicate and duplicate and make more copies and make life reproduce. We found in 2009 that this could happen. And it can be done if the precursor molecules, these would be our precursor molecules, happen to be present, and if they could form. And we found that through Stanley Miller's experiment that these can form. So how do we do this? Well, if we drip solutions of amino acids or RNA nucleotides onto, say, hot sand or clay or rock, they would polymerize. They would form those longer chains. So all that we needed was that hot liquidy soup that was going on in the ocean, and we need to get some of that onto something that's hot, and then we could have polymerization of those. So in order for life to come to fruition, we needed some kind of cell. and we're calling it a protocell because it's a primitive form of a cell, to create a safe, harbored environment for life processes to be able to happen in the first place. And some of those would be reproduction and metabolization. And hopefully you recall from metabolizing things, this is where we get the enzymes that we need for replication. This is where we get our A, T, C, and Gs from. This is where we get our U's from. These nitrogen bases are byproducts from what we eat and what gets broken down through metabolism. So because of this, we do need some kind of a cell to put those in so that we can have self-replication. So what are protocells exactly? Protocells are very similar to vesicles inside modern cells. Recall that vesicles are used kind of as a transportation system at this point for proteins and other products that need to move within the cell or be removed from the cell entirely. They're fluid-filled, and they have a membrane-like structure similar to the lipid bilayer that we have in our cells today. We can also see that protocells are capable of reproducing, and we see that going on in this image right here. We have a larger protocell or vesicle giving rise to more. It also has been recently shown that abiotically formed vesicles can exhibit these same properties of life, including reproduction and metabolism. So these cells right here could metabolize to create an internal environment containing those necessary building blocks of life, those nucleotides. The first genetic material was very much most likely RNA. It was an RNA world at one point. Czech and Altman found that RNA plays a central role in protein synthesis, and I'm hoping that you recall the processes of transcription and translation. RNA can also act as enzymes. Czech called these RNA catalysts ribozymes. Ribozymes catalyze many different reactions. Here we see a ribozyme, which is a strand of RNA, and as you recall, RNA is single-stranded, but it can form these hydrogen bonds with the appropriate base pairs, and this allows for an enzymatic activity to happen within RNA. It can act as an enzyme. These can be self-splicing, so they can cut themselves, and they can also make copies of short segments of their own RNA, so they can be self-replicating. We're going to draw the line for part 1 of chapter 25 right here. We'll continue looking at the fossil record as we move forward into the second installment.